Hey everyone, welcome back to The Explainer. Today we're doing something a little different. We're embarking on a truly fascinating journey, a spatial descent really, shrinking ourselves down to explore the absolute fundamental building block of all life. We're going to zoom all the way from the macro world we interact with every single day, straight down into the hidden microscopic universe that makes every bit of it possible.
Here's our microscopic itinerary for today. We'll start with the basic unit of life, then look at the history of cell theory, explore the wild diversity in size and shape, check out minimalist prokaryotes, dive into compartmentalized eukaryotes, and finally, wrap up with the fluid mosaic membrane.
All right, Section 1, the basic unit of life. What exactly makes us alive?
I mean, look around you right now. You see living things and you see non-living, inanimate things, right? But what is the actual difference? What makes an organism living? What does an animate being have that a rock just doesn't? Well, the universal answer, the big secret, is the cell. Whether you're made of one single cell or trillions of them, anything less than a complete cellular structure just doesn't equal independent life. It is literally the fundamental structural and functional unit of everything alive. Now, to wrap our heads around how this tiny thing pulls off the literal miracle of life, scientists use something called reductionist biology. Sounds fancy, but it basically means using physics and chemistry to understand biology. Take the incredible scientist G.N. Ramachandran, for example. He used heavy-duty math and physics to map out crazy complex protein structures, like the triple helix of collagen. By bridging these fields, he basically proved that at its very core, biology is really just applied chemistry and physics. How cool is that?
Moving right along to Section 2, the history of cell theory, or the blueprint of life.
It's honestly amazing how fast we figured this blueprint out once we had the right microscopes. We didn't get it overnight, but the pieces fell into place really rapidly. In 1838, Matthias Schleiden, a German botanist, realizes all plants are made of cells. Literally the next year, in 1839, Theodor Schwann is looking at animal cells and spots this thin outer layer, what we call the plasma membrane today. So together, they formulate the initial cell theory. But there was a catch. Where did new cells actually come from? Fast forward to 1855, and Rudolf Virchow drops the mic, giving the theory its final shape. He stated, "On the cellula. Basically, all cells come from pre-existing cells. Boom. Theory complete.
Okay, Section 3, diversity in size and shape. Welcome to a universe of extremes.
So, if you have this picture in your head of a standard or uniform cell, just go ahead and toss it out the window right now. The extremes here are wild. Let's look at the microscopic scale. You've got mycoplasmas, the absolute smallest known cells, coming in at a teeny tiny 0.3 micrometers. For context, your typical bacteria are around 3 to 5 micrometers. And human red blood cells, they clock in at exactly 7.0 micrometers in diameter. It's staggering when you realize they are massively different in size, yet they're all playing by the exact same biological rules.
But wait, it gets crazier. We don't just stay in the invisible microscopic world. Do you know what the largest isolated single cell is? It's an ostrich egg. Seriously, a single, massive cell that you can literally hold in both your hands. Thinking about this huge outlier perfectly contrasts the microscopic scale we just looked at, and it just proves how incredibly adaptable cellular life really is.
And hey, the diversity isn't just about size, it extends entirely into their shape too. In biology, form strictly follows function, always. So you have these round, squishy, biconcave red blood cells made to squeeze through tight capillaries. And you've got white blood cells acting like amoebas, literally changing shape to gobble up invaders. My absolute favorite, though, are the nerve cells. Because they have to shoot signals across vast distances in your body, they're thread-like and branch out extensively, making them some of the longest cells we have.
Let's zoom in a bit more. Section 4, prokaryotes. The minimalist cells, living life without compartments.
Think of these guys, like bacteria and blue-green algae, as the rugged off-roaders of the biological world. They function perfectly without any fancy compartments. Their DNA, it's totally naked, just hanging out, no nuclear membrane locking it safely away. A lot of them also carry these cool little circular rings of extra DNA, called plasmids, and these are a real game changer because they can grant special traits, like antibiotic resistance. They use these tiny 70S ribosomes as their dedicated little manufacturing machines. But honestly, the wildest part is their outer armor. Inside, they're minimalists, sure, but outside, they have a tough, tightly bound, 3-layered cell envelope. It starts with a sticky glycocalyx, then a sturdy cell wall, and finally, a plasma membrane. 3 distinct layers acting together as one incredibly tough protective unit.
Moving right along to Section 5, eukaryotes, the compartmentalized cells, or what I like to call the mansions of the microworld.
Right, so if prokaryotes are the tiny minimalist studios of biology, eukaryotes are the sprawling mansions. We're talking protists, plants, animals, and fungi. The architectural difference is night and day. Where prokaryotes leave their DNA naked and lack membrane-bound organelles, eukaryotes are absolutely obsessed with compartmentalization. They have an organized, secure nucleus protected by a strict nuclear envelope, and their cytoplasm is packed with dedicated, membrane-bound organelles. They effectively have specific rooms for specific cellular activities.
But, even within these fancy eukaryotic mansions, the interior design totally changes depending on the organism. Take a look at plant versus animal cells. A plant cell is like a fortress. It's got a thick, rigid cell wall, a large central vacuole, and plastids, like chloroplasts, so it can actually make food using sunlight. Animal cells? Nope, no cell wall, no plastids. Instead, they rock these things called centrioles, which play a major role in cell division. So they use the same foundational blueprint, but customize it beautifully for their highly specific survival needs.
All right, down to our final zoom. Section 6, the fluid mosaic membrane, the ultimate bouncer.
We are now initiating our deepest zoom, right to the absolute edge of the cell, staring at its selectively permeable plasma membrane. Specifically, we're exploring the fluid mosaic model, a brilliant concept proposed by Singer and Nicholson back in 1972. The mechanics here are just so smart. It is not some rigid, solid wall. The fluid part refers to the quasi-fluid nature of the lipids. It actually allows proteins to float and move laterally across the overall structure, almost like icebergs in a sea. It's flexible, it's dynamic, and that fluidity is utterly essential if the cell wants to grow, secrete things, or divide.
So what's this dynamic border control actually made of? Well, its architecture relies on a phospholipid bilayer. Imagine these molecules with polar heads facing the outside watery environment and hydrophobic, water-fearing tails hiding safely on the inside. Floating around in this lipid sea, you've got cholesterol, along with both integral and peripheral proteins and some carbohydrates. This specific chemical cocktail is what allows the membrane to act as a highly selective bouncer. It dictates exactly what gets a free pass, like water slipping through via osmosis or neutral solutes through passive diffusion, and what gets strictly kept out.
So as we wrap up this explainer and zoom all the way back out from the microscopic world to the macro scale of your everyday life, I want to leave you with this provocative thought. How does understanding this tiny, squishy lipid bilayer change how you view gross physiological processes like your own memory, your digestion, or your immune defense systems? I mean, every single major function your body performs is, at its core, completely reliant on the integrity and the microscopic chemical transactions happening at the very edges of these cells. It kind of changes your perspective on what it means to be alive, doesn't it? Thank you so much for exploring the basic unit of life with me. Keep learning and stay curious.